Our next speaker actually will talk along the same vein about what we're doing with Helm and also our biological registration. So, introducing Roland.
Hi, everyone. Just between the coffee break, it's this little presentation on biologics at ChemAxon. ChemAxon you will traditionally know as a cheminformatics company. So you may ask yourself, like, what the hell are these guys doing with biologics? Sergio has given you quite a nice overview about where the challenges are, and the challenges actually go into a similar fashion where traditional bioinformatics actually don't meet the expectations anymore because you have so many modifications on your biological macromolecules, you need to represent them somehow. At the same time, the molecular graph is just not the way how to move forward with these large biomolecules. There was a communication sideways just to highlighting the importance of biologics with Alex Driver and one of the potential guests to this UGM, and he apologized, stating that I won't come, but if your company were called Bioaxon, I'd be there in an instant. Now, we're certainly not going to change the name, but albeit just a subjective answer to the question how important biologics are, a second fact I'd like to pull up is from Doug's presentation, stating that in the year 2012, 26 out of 39 therapeutics approved by the FDA were small molecules. Well, it sounds like a good number. But at the same time, it means that about 1/3 of these molecules are biologics, right? And biologics contribute more and more to the therapeutical development in medication. So let me try to get back to where biology has already been involved for quite a while in ChemAxon tools, and probably the most relevant user exposure has Marvin's sketch towards the end user. And I'd like to highlight now a few things which we've implemented in the recent version 6.0 and give a short outlook about where our developments are going with respect to Marvin's sketch in the representation and the handling of biomolecules. So for example, in Marvin's sketch 6.0, a new feature is that we are now able to import sequences in FASTA format. It's the predominant exchange format in biological sequence information. You can either give the information whether you are loading a sequence as DNA, RNA, or peptide molecule. But we also have a sort of automated sequence recognition system which allows you, with a certain, limitation of ambiguity, try to guess intelligently what sorts of sequence you're actually encountering when you just pipe in your FASTA format. The same thing of conversion is actually available also from the import source dialog in Marvin's sketch. And what happens then is you'll get a nice cleaned up sequence. We have these snake-like line breaks in longer peptide sequences which don't fit on the screen in order to just somehow condense the view of the molecules a little bit. The technicality is that, you see the letters here on the canvas for the individual amino acids. They are still chemical information, so they are these superatom groups, and behind each of these superatom groups, the actual full molecular structure is still available, and you can just expand it and recondense it in order to have additional reduction in complexity in displaying such a biomolecule. A new feature related to that is that we have started to implement an unlimited number of so-called attachment points. Now, what's the use of these attachment points? When you have the sequence represented on your canvas in a condensed way, you are still able to use your chemical drawing tools in order to add bonds to these superatoms. Now, where do these bonds go? These bonds do go to the actual attachment points of the superatom groups. And I've picked out an example where it might be relevant for a biological use case, L-histidine, for example. L-histidine has possible modifications. Phosphorylation may occur, for example, in the 1 nitrogen or in the 3 nitrogen. And the regular template which comes with Marvin's sketch has only 3 attachment points. Now, what do? You can just define a 4th attachment point you can define attachment points at any given atom of the structure if this is somehow chemically feasible for you. So that's something we've implemented here. You just right-click on the corresponding atom, add your S-group attachment point, and still all of the kind of information about how to condense the structure back into the single residue abbreviation is still available. Things which are new features and will be available in future versions 6.1 or 6.2 are dealing with the representation of biomolecules. So one example here is how do we represent sequence bridges, cyclizations of peptide chains, for example. Well, at the moment, it looks pretty ugly. So we are working on implementing these sorts of angular bonds in order to be able to represent cysteine disulfide bridges or these cyclizations. We are implementing a system which enables you to have persistent so-called leaving groups. So in the previous slide, I've shown you about these attachment points of the abbreviated residues. Now, obviously, in the standard mode, you may want to see the actual chemical structure, a correct chemical structure of your amino acids. Now, whenever you bring two of these amino acid residues together in a single chain, there are some atoms leaving in order to make the proper connection. And what is currently not implemented is that these sorts of informations about the leaving groups are in fact persistent. And whenever you have drawn such a peptide chain and you delete a bond in the middle of the sequence, the leaving groups are currently not regenerated. And that's something we are working on in future versions. Last but not least, we are talking about high-quality structure drawing. One of the weaknesses of Marvin Sketch currently still is that whenever you expand individual residues of a chain, the cleaning mechanism is, well, you can see it here. The carbonyl oxygen is overlapping with the residues in the sequence. So we do make some horizontal space for the expanded residue, but I guess we still should improve how we increase the space for the molecules in the vertical position. After all, biomolecules are large, right? From one perspective, now you can draw molecules. That is nice. The scientist can enter something. He can visualize a molecule. But after all, there's a lot of information related to these biomolecules, and you would like to somehow interrogate maybe large sets of biomolecules. So you need some sort of information management. And the beginning of such information management for us at ChemAxon is, here you see a screenshot of our new compound registration product. It starts with the registration of some information. Small molecules, small molecule structures, large molecules, well, what do you register there? There are a few challenges related to biologics registration. And let's just talk about large molecules, everything which has some sort of a chemical structure associated to it. You can briefly group it into three different aspects. So one of the things is the structure itself. The structure itself, because it's large. There are lack of representation standards for kind of capturing the additional chemistry associated with the pure sequences. And actually, Sergio has given a really nice overview about the reasons and all these representation issues which are arising there. The second thing is input methods. We also talked about it, that you probably do not want to draw such a large molecule with a chemical sketcher. At the same time, a pure text editor, in order to edit your sequence, well, how do you add your chemistry information there? So the second part is about annotation. Annotation in terms of registration usually carries very important bits of information which make a distinction between molecules which may share the same structure, actually. A reason for it is, for example, that patented are usually not large biomolecules themselves, but cell lines which are used in order to produce these biomolecules. When these cell lines are patented, the somehow barrier of copying the molecules are much higher, because you have to undergo the process of showing that the protein you developed in another cell line is actually behaving in the same way in your body as the original molecule. So The second part here, in terms of annotation, deals with domain-dependent conventions for describing certain aspects of your biomolecule. One expert research group may actually term certain things in a very different way than other expert research groups. And this creates confusion whenever you would like to bring this information together in a consistent fashion. This deals almost very tightly with aspects of the business logic in order to register these biologics. So a lot of the things you try to register are not only determined by the structure alone, but they are also determined by the additional descriptors. How was it produced? What's the genealogy behind the molecules? And last but not least, you always have to make sure that in order to describe these processes properly, you have to kind of implement a way of coping with a very, very high flexibility in the underlying workflows, because state-of-the-art technology is constantly evolving and changing the landscape how biomolecules are actually produced. Now, we can offer a few solutions to the problems I've just listed here. In the long-term aspect, we are trying to deliver you a biologics education system which is able to cope with at least most of these aspects in one way or the other. The lack of representation standards, to a large extent, and with respect to the limitation, everything which currently can be described 100% accurately can be resolved by Helm. We are anticipating that Helm will evolve, so we actually see a great chance that Helm may evolve into somehow being able to describe certain ambiguities which are of relevance for the drug discovery industry when it comes to the development of biotherapeutics. Input methods, well, there needs to be some sort of new tool, BioSketcher, call it, which kind of integrates chemistry together with sequence information. The Helm editor is a viable first approach in this direction, but we've experienced it has some limitations, some flaws in dealing with these molecules. And we believe it can be made much more user-friendly than it currently is. Distinct descriptors, well, in the end, it's all about the data model. And if you apply something which is agnostic towards the type of entity you're going to register, you may have a good starting point to be somehow warned against anything which may show up in the future in terms of let it be something which is it's kind of built from components which are already known, but as a total are a completely new entity which nobody has ever thought about before. Domain-dependent conventions, well, in the very easiest case, they can be solved with synonym tables, just indicating, okay, this means something in this group and this means something in the other group. Depending on the user logging onto the system, he may be presented the information which is preferable for this expert domain. In the more complicated cases, this will largely go into the area of ontologies, and we are seriously considering and getting partners in these sorts of ontology domains. With respect to the business logic, well, integrated registration means that you need to account for the data from a large variety of sources. And from ChemAxon's perspective, of course, we can deliver this ourselves, but then we have a long way ahead of us. But there are systems which are currently in place in a lot of the companies we are actually communicating with. And they have certain specialties and expertises and best practices which we don't want to really interrupt. So in this context, we will develop certain bits and pieces there in the long term, but we will try to use as much as possible of what you have already organized in this sort of genealogy context. And probably the most difficult part here is being able to actually register workflows in a way that you are registering biological entities in a fashion that you capture all relevant information and you capture also all information about how this biological entity is actually produced and developed, no matter what sorts of experimental steps are in between. And all these processes, of course, are highly parallel usually. So there's an additional level of complexity in this. All right, let's switch to somehow a closer view. Where do we want to go from now, just in a few months, maybe. Let's strip off certain things which appear to be a little bit out of scope in the context of that we are adopting Helm as a biomolecule registration standard. And combined with the fact that, well, ChemAxon, cheminformatics company, we are somehow having a compound registration. Would be nice to have a module for biomacromolecule registration, because that's right closely related to each other. So these are the three areas we are focusing in right now at the moment. And there's just one tiny little change. So BioSketcher is neat, but it also needs capacity to develop such a thing. So we will probably go first in the alpha versions of a macromolecule registration with the existing Helm editor, improve it where it's necessary in order to make it usable for the end user. But we anticipate at the same time that probably the editor may not be the preferred way of interacting with a toolkit which deals with registration, but it will rather be a set of services in the backend which do some batch processing of the data which is coming in. Sequences are automatically converted to Helm, which then get registered in the biomolecule database. You may use the editor from the scientist perspective in order to add your modifications, but often these modifications are already present in databases in-house, just the annotation formats may differ, and we have to worry about rather how to bring these automated annotations, transfer this to a proper Helm notation, and then update the registration records for the entities we are looking at. Again, myself, stepping aside from being a ChemAxon member, our contribution, Sergio highlighted it and actually surprised us quite well with this nice little prize for it. So our contribution was that the tools which previously had been production tools at Pfizer were freed of the chains of Pfizer infrastructure in order to be able to provide it to the open source community. And the reason why we did this is actually twofold. First of all, we believe in the principles of Pistoia. We are a company which is trying to bring you solutions, right? We don't care about competitors, but we want you to enable certain new technologies for your own benefit. The second reason is that looking at Helm, what it can do in its current state and where it's promised to go, we believe it's a really viable approach in the border between cheminformatics and bioinformatics. I would even term it biocheminformatics for the sake of giving it a name. But going back to ChemAxon now, where do we want to go actually in terms of biomacromolecular registration? I've put up a brief schema here about how we imagine an architecture of a very, very simple biomolecule registration system. It will be based on the open source Helm editor annotation toolkit. Parts of the functionality are exposed as a public API. And since we've created an applet out of the Helm editor, we can actually make it available in a browser. And that comes in a similar context as our compound registration system, which is a browser-based web application. On this API, we built a set of core web services which deal with the registration. And behind these web services, we have a biomolecule database, which stores the monomer information you've heard about, relevant for being able to chemically interpret a Helm notation, and to store the actual biomolecules you are going to register. In the second phase, we are looking at auxiliary web services, which will enable your scientists to do some more informed tasks on the entity you have actually registered. So Sergio mentioned the context of having natural analog sequences, which are very well necessary in order to do blast searches, for example, because blast searches are chemically unaware. There may be certain calculator services which calculate properties of biomolecules, Miller extinction coefficient, for example, just as a very simple example. We may think about integrating other information to the biomolecules which is relevant. Three-dimensional structure, for example, from your crystallization screens. We can map the coordinates onto the chemical atoms of the biomolecule structure itself. And last but not least, in the context of antibody-drug conjugates, you are looking at something which is partly biological, partly chemical. Now, many organizations already have a small compound registration system, right? So our aim is to, first of all, be able to integrate with compound registration, obviously our own, but also compound registration systems existing at different organizations already. It is, after all, just a sort of service that brings in the corresponding compound ID and makes the system known that, okay, this is a biologic, here's my antibody molecule, and this is bound to something I have registered in my small molecule library. In terms of desktop applications and end-user applications, I don't want to waste too much time on this slide. I think the core point here is that Instant JChem, to a large extent, is based on core technologies of ChemAxon. And these core technologies we are aiming to extend also in the context of biomolecule handling, data representation, and such. So where you have JChem and Marvin modules and further calculators, you may also find a BioCore API and certain web services which can be exposed directly as the core technology for end-user applications such as Instant JChem. So you may find yourself in a situation where you have tables where instead of a chemical structure, you see a biological structure, and you have a few calculated fields based on biomolecules. And the kind of development timeline for us is that we'd like to release an alpha version in kind of summer 2013, which is based on, well, the Helm notation itself. From the Helm notation, we store the strings in a biomolecule database, which has a set of web services. Both of them, the major protocols, SOAP and REST services, are supported by this. The alpha version may still have some performance constraints, so we for sure have to make optimizations in terms of, well, data interrogation and searches on this data. But however, we'd like to kind of encourage you to try out if biologics at your organization are an issue and you're struggling with the data representation or the data handling of biomolecules. I think this is a proper time to somehow initiate discussions on where should the development after the toolkit level actually go. With respect to a timeline, so I mentioned the Biomolecule Toolkit. We hope to have something like a biomolecule registration system as a backend technology, potentially still without a proper user interface, but this is something which is going to be available in later stages of the development. And we are also looking at, in the context of proper genealogy handling, about biomaterial registration. And with this, I'd like to conclude my presentation here, and I'm open for questions.
I'm really happy to see that the apartheid of small versus biological molecules is about to become history. But nevertheless, some concrete issues, such, for example, support for the MOL2 format. We had a horrible bad time trying ChemAxon to read more readable MOL2 and the other way around. Now, we don't know who's the bad guy in the story, but the two softwares don't discuss to each other, even though the standard format should be standard. And the other question is, out of the existing ChemAxon tools, how many and to what extent will be biomol compatible? You know, it would be cool to let the structure checker loose on a PDB file and get your sick ligand fixed and the protein protonated at the same time, and force field types assigned. There's a lot of things you can do in this direction.
Regarding the first question, standards to follow, since we are pretty much engaged in this Helm project, for us, we also see that the preferred data exchange format will be the Helm notation itself. If this is not appropriate because of things we implement and, well, the kind of timeline of the Helm evolution doesn't cope with it, then we have to think about different alternatives, obviously. But that is something which is, at the current state, still unsolved. So it will be an entirely new format anyways in this case. With respect to the second question, to some extent, I would say that I'm sorry, can you repeat that? I must have lost it on the way.
So out of the existing small molecule tools, will you
Yes,
expand them
okay.
to incorporate to make
will-
them biomol compatible?
It will certainly need some tweaks and optimizations. Think about 2D clean algorithms, for example. It has to somehow realize that it is looking at a biomolecule and then expand or clean the backbone accordingly, so that it's not having a kind of sequence cleanup where the backbone is going all around the canvas. There are a few other optimizations also in other tools available for sure, but we are still exploring which of these things actually work out of the box and which need some customizations and context, but it's on the agenda.